AS Level Biology 9700, Paper 1, Variant 1, May, June 2025. Question number 14 to 26. And in first video, we had discussed the question number 1 to question number 13. So question number 14 is, which statement explains why large volumes of water maintain a constant temperature when the air temperature increases? So in that case, when the air temperature increases, the large volume of water is maintained. You can say maintain a constant temperature. So four options are given over here. The latent heat of vaporization of water is high. So latent heat of vaporization is the amount of energy which is needed to change the liquid into gaseous form. So here this property explains, we can say that the sweating is an effective cooling mechanism, or a lot of energy is required to change water from a liquid into a gas, but it does not directly explain why a large volume of air maintains a stable temperature. So when the air warms up, but they are not telling us how it takes place. So this is not a correct option. So we go with the second option, and second option is the specific heat capacity of water is high. So the specific heat capacity of water is high. So I would like to say that water has a high specific heat capacity. It is correct, meaning it can absorb a large amount of heat energy with only a small increase in temperature. So this is very important property of the water because it has a specific heat capacity. So it can absorb a large quantity of heat into it. So this property allows large bodies of water, large bodies of water like oceans, lakes, to maintain a relatively constant temperature even when the surrounding air temperature fluctuates significantly. So the water absorbs the excess heat from the atmosphere, effectively acting as a heat sink. So this is the correct option. C, we will move towards the C. Each water molecule can form a hydrogen bond with a maximum of 3 water molecules. Each water molecule can form a hydrogen bond. While hydrogen bonding is a fundamental property of water that contributes to its high specific heat capacity, the statement itself is not the direct explanation for the phenomena described, no doubt. And it can happen, but this is it is explaining the structure of the water molecule, not the property of the water molecule that it can absorb large quantity of heat. So this is not the correct option. Water is able to evaporate rapidly from a surface with little loss of heat. So this is the water is able to evaporate rapidly. So this statement is incorrect. This is not a correct statement because evaporation is a cooling process. It involves the removal of high energy molecules from the liquid surface, so leading to a significant loss of heat from the remaining liquid, but it is not explaining that the how the water can maintain a constant temperature. So in this way, A, C, and D are not the correct options. Only B is the correct statement. Question number 15, and question number 15 is, which statements about the mode of action of an enzyme are correct? So mode of action of enzymes are correct. Some enzymes have a region on their surface to which the substrate has a complementary shape. They are talking about the active sites on which a substrate is attached. So enzymes have a specific region or which is active site. So according to this, the statement is correct. And second statement is some enzymes and their substrates can change shape slightly as the substrate enters the active site. This statement is also correct according to the induced fit model. So, induced fit model can explain this statement, that is when there is when the substrate is attached on the enzyme, so the active site of the enzyme and the substrate can undergo a slight conformational change to achieve a better fit, so when they bind, optimizing the reaction. So this statement is also correct. Now moving towards the third statement, enzymes permanently change shape when they react with their substrates to form an enzyme substrate complex. This statement is not correct. Enzymes are, you can say, the catalyst, meaning they speed up the chemical reaction without being permanently or altered or consumed in the process. They can be changed, they can be used again and again. So in this way, a very small quantity of enzyme is used for the process for the formation of enzyme substrate complex. So in this way, they cannot attain a permanent shape. So in this way, they will be wasted. So this is not the correct statement. Only statement number 1 and 2 are correct. So according to this, A is not a correct option, C is not a correct option, D is not a correct option. Only 2 statements are correct. So according to this, B is the correct option. Now moving towards the question number 16. And question number 16 is, they have given us a graph. The graph shows the effect of increasing substrate concentration on the rate of reaction of two enzymes that catalyze reactions with the same substrate. So they have shown us rate of reaction on y-axis and substrate concentration on x-axis. So you can see the enzyme P and enzyme X. So Q. So the four statements, enzyme P, rate of reaction is different from enzyme Q. So enzyme Q has a higher Michael-Mentons or Km value and a higher affinity for the substrate than enzyme P. So you know that Michaelis constant is the substrate concentration at which the reaction rate is half of the maximum velocity. So lower Km indicates that the enzyme reaches half of its maximum reaction rate at a lower substrate concentration. So this means the enzyme has a higher affinity for its substance. So it can bind and catalyze the reaction more effectively when the substrate is scarce or less. So enzyme Q has a higher Km and higher affinity for the substrate than QP. If it has higher Km value, it means it has less affinity. So this statement is not correct. Then enzyme Q has a higher Km and a lower affinity for the substrate than enzyme P. Higher Km and a lower. You can say this statement is again not correct. Then enzyme has a lower Km and higher affinity for the substrate than enzyme P. Yes, this is the correct statement. That is, it has lower Km. It has lower Km and higher affinity. So that is the reason lower Km means it has higher affinity for the substrate than enzyme P, because enzyme P has higher Km value. So enzyme Q has a lower Km value and a lower affinity. Now again, this statement is not correct because it has lower Km value means it has higher affinity for the substrate than enzyme P. So according to this, only C is the correct option. Because you know enzyme Q reaches its maximum rate of reaction at a lower substrate concentration. You can see this is the lower substrate concentration as comparison to P. P is taking too much substrate concentration, whereas it has lower substrate concentration. You can see in this way, this, and it is taking this. So enzyme Q has a maximum rate of reaction. So its reaction is fast. It has lower substrate concentration as comparison to enzyme P. So this is evident from the graph where the curve for enzyme Q flattens out much sooner than the curve for enzyme P. So therefore, enzyme Q has a lower Km. So then a lower Km value signifies, or we can say that has a higher substrate affinity, has a higher affinity for the substrate than enzyme P. So according to this C is correct option. Next question number 17 is there, and the cell surface membrane structure is described as a fluid mosaic. Fluid, a liquid or jelly-like, and mosaic when something is attached over it. So the fluid mosaic model describes the structure of the cell membrane, and the fluid part refers to the phospholipid bilayer. You can see, you know that a phospholipid bilayer is formed in case of cell membrane, and then the molecules are constantly moving, allowing the membrane to be flexible. So mosaic parts refers to the various components which are scattered or which are present throughout the uh, layer, so creating a pattern similar to the mosaic tile artwork, and you can say it is embedded. These different types of proteins, you know, which are attached over there on this cell surface membrane. So the first option, which correctly describe the mosaic part. So the different patterns that are obtained by the moving phospholipid molecules. So different patterns. The different patterns are not actually explaining the correct shape or correct structure of the this fluid mosaic, because the different pattern, there is not different pattern, actually the different proteins are arranged over the phospholipid bilayer. The random distribution, so this is not the correct option. Second, the random distribution of cholesterol molecules within the phospholipid bilayer. Cholesterol molecules help in the, you can say, not in the random distribute mosaic part, actually they help in the fluidity of the cell surface membrane. So this is not correct option. The regular pattern produced by the phospholipid heads and membrane proteins. So this is the arrangement of phospholipid bilayer, not the mosaic. So the scattering of the different proteins within the phospholipid bilayer, yes, because they are scattered over the phospholipid bilayer. They are arranged, they are attached over there. So in this way, D is the correct option. Question number 18. And question number 18 is, a student immersed a plant tissue in solution S. Solution S. For the next 30 minutes, the student used a microscope to observe the cells in the plant tissue. So it was immersed for about 30 minutes. During this time, the cytoplasm of the cell shrunk and no longer pushed up against the cell wall. In many cells, a space appeared between the cytoplasm and the cell wall. So which statement are correct? Cell shrunk and no longer pushed up against the cell wall. So in this way, I think it was shrunk. and no longer pushed up against the cell wall. So it can be the shape. So this shape. So which statement are correct conclusions from these observations? So the water potential in the solution S was more negative than the water potential in the cytoplasm of the cells at the start of the experiment. So it means when this cell was kept into solution S, the water moved out of the cell. Why it moved off the cell? Because the external solution has a lower water potential as comparison to cell cytoplasm. So that is the reason the water moved out of it. So it is shrinked. So the first statement is correct. Without a cell wall to provide support, it is likely that many of the cells would have burst. The cells do not burst. The cells do not burst. So statement 2 is not correct. The cell clear space between the cell wall and the cytoplasm of many of the cells was filled with air. It is not filled with the air. It is filled with that solution in which it was kept. So according to this is not a correct option. So space that appears is not, there will not be the air, there will be the solution in which solution is. It will be, there will be solution S. So according to this, only one statement is correct, which is statement number 1. So according to this, the option B is the correct option. Now moving towards the question number 19, and question number 19 is the sample of 500 cells, the sample of 500 cells dividing by mitosis was examined to identify the stage of mitosis for each cell. The table shows the results. Mitosis, a cell division in which chromosome number is maintained generation after generation. It remains the same in the daughter cells. So different stages of mitosis, percentage of cells in the stage late prophase is 23%, end of the metaphase 45%. So between the start of anaphase, it is 24%. Which row is correct? So, so they have told us about 23%, 45%, and 24%. So we have to find out the percentages. Number of the cells, which row is correct? Number of the cells with chromosomes at the equator of the cell. So it means there can be the end of the metaphase. So at the end of the metaphase, the chromosomes arrange themselves at the equator. So this is 45%. 45%, and we have to find out the number. We have to calculate the number from this percentage. So the second one is number of cells with separated chromosomes. Separated chromosome, this describes the cells in anaphase and telophase. So it means during anaphase, the sister chromatids separate and move to the opposite poles. So this process continues in telophase until the cell is divided. So the table provides the percentages of cell between the start of the anaphase and end of the telophase, which is 24. So now we are going to find out the number. To find out the number, we have to 45 and the sample. we will do. So according to this, you can calculate this with the help of a calculator. We will multiply 45 with 500 and we will divide it with 100. So it will be equal to 225. And then second one is 24. So 24 into 500 and divided by 100. Then we will multiply 24 with 500 divided by 100. So it will be equal to 120. So this is going with A. 225 will be the number of cells with chromosomes at the equator of the cells. Number of cells at equator. And 120 will be the number of cells with chromatids. So A option is the correct option. Now we will move with the next question, which is 20. Question number 20 is, the diagram shows a chromosome. A chromosome, what are structures P and Q? Very simple question in which they have given us a diagram in which they have asked what is P. P is centromere. With which the chromatids are attached. So Q is the part of the chromatid or end of the chromatid, which is called as telomere. And you know, telomeres are the protective caps. They prevent the loss of genetic information during DNA replication. So they protects genetic information. During replication. So P is the centromere and Q is the telomere. So according to this, A option is not correct, D option is not correct. So B or C can be the, so B is the correct option. So moving towards the next question, and next question is, which processes are used by stem cells during tissue repair? First of all, tissue repairing, you know, the mitosis takes place when the tissue repair occurs. So stem cells use all these three processes. Cytokinesis, when cytoplasm, after the completion of mitosis, the cytoplasm, after telophase, the cytoplasm is divided. So this is cytokinesis. So it happens, it takes place when there the tissue repair, because the cell has to be divided into new cells. Then second is DNA replication. So DNA replication is the process of making an exact copy of DNA cell. So this is the important. So cell divides so that each new daughter cell receives a complete set of genetic instructions. If DNA replication does not take place, then this cell's chromosome number will not be equal. The cell will not maintain their chromosome number after mitosis. So this DNA replication also takes place. Then third is transcription. Transcription is the process of, you can say, the first stage of protein synthesis, in which during this process of transcription, there is a creation of RNA molecule from a DNA template. So this is the first step in gene expression. So it allows the stem cells to produce the specific proteins needed to differentiate into specialized cells. So according to this, A is the correct option. B, C, D. Because all these three processes are used by stem cells during tissue repair. Question number 22. And question number 22 is the enzyme telomerase. The enzyme telomerase prevents loss of telomeres after many mitotic cell cycles, which cells transcribe a high concentration of telomerase. So they have given us neutrophils. First of all, telomerase. Telomerase, the enzyme. Telomeres, protective caps, which protects the genetic information during the mitotic cell cycles. So neutrophils are the type of the white blood cells. Neutrophils are the white of the cells that have a short lifespan. So they have a short lifespan. So according to this, and the high rate of production requires telomerase to maintain the telomeres of the progenitor cells. So neutrophils are not those cells. Mature red blood cells. Mature red blood cells, they don't have nucleus and DNA, so they cannot transcribe telomerase or any other protein. Simple. Third, activated memory T lymphocytes. So they are also the type of white blood cells that can rapidly proliferate in response to a pathogen they have previously encountered. So this rapid and extensive division has high telomerase activity to prevent telomere shortening and allow the cells to form a large population. Therefore, T lymphocytes are the correct cells. So, activated memory cells are the correct. So, according to this, D is the correct option. A, B, C are not the correct options. Question number 23. And question number 23 is, which statements are correct? All polypeptides are coded for by genes. Polypeptides, yes. This statement is correct because polypeptides are coded by genes, so some can be synthesized by non-ribosomal peptides. So we can say this is the correct option. All genes are made up of a sequence of nucleotide. Again, this is the correct option, a small segment of DNA. So all gene mutations result in a change to the nucleotide sequence. So statement 3 is also correct because a mutation is defined as change in the DNA sequence. So this change can be a single nucleotide substitution, an insertion or a deletion. So according to this, this is the correct statement. All gene mutations will result in an altered polypeptide. No, all gene mutations will not result. So there are several types of mutations that do not lead to a change in the polypeptide. So in this way, this statement is not correct. So according to this, B is the first three statements are correct. So B is the correct statement. Now question number 24. A double-stranded DNA molecule was analyzed and 29% of its nucleotide bases were found to be adenine. 29% adenine. So which percentage of its nucleotide bases will be cytosine? So, students, the adenine is always equal to the amount of thymine. Adenine always pairs with thymine. So guanine will pair with the cytosine. So if adenine is 29%, so thymine will also be 29%. So the remaining percentage will be of guanine and cytosine. So 29 plus 21 will be equal to 50, and this 25 and this there will be 29 plus 21 will be 50. So according to this, A, because we have to cytosine, we have to tell the, so according to this, A is the correct option. B is 29%. So cytosine is not equal to the, it will not bond with the adenine. So it is not. 42%, again, no. 58%. So all these percentages are wrong. Only A is the correct option, which is 21%, because it will be equal to, because it will bond with the guanine and cytosine with one another. Question number 25. And question number 25 is, which row correctly states the name of the known coding sequence of RNA and the type of cell in which the non-coding sequence are removed? Name of the non-coding sequence, exon and introns. Type of the cells in which non-coding sequence are removed, they are eukaryotic or prokaryotic. So, non-coding sequence after transcription, when they are removed after transcription, they are called as introns. So, non introns. So this process is called as RNA splicing. So prokaryotic cells will lack introns in their genes, so they do not perform RNA splicing. So therefore, the non-coding sequence are introns, and the process of removing them occurs in eukaryotic cells. So they are not in prokaryotic cells. They are not exons, they are introns. So according to this, C is the correct option, because the introns are the non-coding sequence. And next question 26 is, Water potentials were measured inside a leaf cell. Water potentials. So in the air spaces of the leaf, inside a leaf cell, in the air spaces of the leaf, and in the, so these are the three locations where the water potentials will be measured. So inside the leaf cells, which value of the water potential would allow water to move from the leaf to the atmosphere? H2O from the water towards the in the atmosphere. So it means the water moves from an area of higher water potential to an area of lower water potential. So water to move from a leaf cell to the atmosphere, the water potential must be highest inside the leaf cell and the lowest in the atmosphere. So creating a steep gradient. So inside the leaf cell is -1 and airspace -7 and the atmosphere -100. -100 means less water potential as comparison to leaf cells. So this A option can be the correct option, but here because only way when the water will move from its higher water potential towards the lower. So when we go with the second option in which showing the -100 means the lowest water potential inside the leaf. So water will not move into the atmosphere. Again 1 and 100. So only and 100, 1 and 1. So this is not the only. A is the correct option in which the water potential is high as comparison to atmosphere. So what due to which the water will leave the leaf and it will move into the atmosphere because the atmosphere has lowest water potential which is -100. So in this way, A is the correct option. So students, it was all about question number 14 and 26. Now wait for the third video in which I will complete this paper, that is 27 to 40. So, but please don't forget to like, share and subscribe my channel. So it will help you to find more papers. And thanks for watching.